A very warm welcome to the participants of the Proteomics Advanced Winter School 2021. Presented by Proteomics Lab, Indian Institute of Technology, Bombay. Day 6, Quantitative Proteomics, Label-Free. Lecture 2, Applications of MS-based Proteomics.
Gentlemen, it is our great privilege and honor to have amongst us one of the pioneers in the field of proteomics research, Professor Bernhard Koester. He joins us from Technical University of Munich, Germany. Professor Bernhard Koester obtained a PhD in biochemistry from the University of Oxford. Thereafter, he completed his postdoctoral training, first with Professor Matthias Mann at EMBL, Heidelberg, Germany, and at the University of Southern Denmark. Professor Koester is the chair of the Technical University of Munich Department of Biosciences and a member of the Graduate School Experimental Medicine. His research is funded by the Center for Integrated Protein Science, Munich, the German Consortium for Translational Cancer Research, and also the pharmaceutical industry. Between the years 2000 and 2007, Dr. Koester worked for the biotechnology firm Cellzone, where he held several positions, including that of Vice President of Analytical Sciences and Informatics. In the year 2007, he was appointed full professor and chair of proteomics and bioanalytics at Technical University of Munich, Germany. Presently, he is the head of the Department for Biosciences and a co-founder and co-director of the Bavarian Center for Biomolecular Mass Spectrometry. Dr. Koester is a member of editorial and scientific advisory boards of several journals, university institutes, and companies. His current research interests include functional proteomics, chemical biology, cancer, biomarker, and drug discovery. Please join me in welcoming Professor Bernhard Koester.
To present a few stories from my team who's been doing all the work, and it's fun to have all these great young people around me that basically as a team accomplish what we do. But before I start, I should make a few quick disclosures. I founded two companies. I've got nothing to do with their daily business, but I'm a founder and shareholder. We do currently get some research funding from Merck, and IBM, Thermo Fisher, and SAP are supporting us in kind with some of our technologies that we build and run. So that has hopefully nothing to do with the content of the science other than it makes it better. All right, so just as a background from my lab, we basically do kind of three things. And one is to map out proteomes, because I think that is an important area just to know which genes actually exist as proteins, where they are in an organism and in what quantities and how they are modified, and so that we might better understand what they do. The second pillar of the lab is actually understanding how drugs work, with a particular view to precision in medicine and oncology. And the third pillar is building proteome tools. And this is really the content of my talk today. And I thought for a workshop on proteomics, this is more fitting than giving you a high-level story about human or plant proteomes, but actually get down to the dirty details of every day's lab business and what we could do there, because it's true to say, I think still, that proteomics is the technology-driven area, and we haven't seen the end of these areas yet. So what I'm going to talk about is one thing in quite some detail, which is proteome analysis by micro-LC MSMS. That's an area that I find extremely exciting at the moment. And the second is to look at FFPE material analysis using nano LC devices coupled to FAMES and MSMS. And I'm just going to hint briefly on an informatics tool that we've built called Prosit, but you're going to have a full talk about Prosit or our IT tools on Friday when Matthias Wilhelm is going to talk about this. So straight in, one of, let's say, the most exciting technology developments in my lab, at least, has been driven particularly by Yang Bian and also assisted by Florian Bayer, where we basically took a look at solving all the problems with nanoscale liquid chromatography, because many of you will know that this comes with a lot of issues. And inspired by the work of other people, in fact, we've started playing around with the idea of using 1 millimeter micro LC columns for some simple reason that you wouldn't worry too much about how much you need to load, and the chromatographic performance should actually be great. And what a lot of people in our area forget is the mass spectrometer can be a great device, of course, but if we do poor chromatography at the front end of this mass spectrometer, it's not going to be able to compensate for all of the mistakes we've made already. But just to put some perspective as to how that sits in terms of scale, the typical nanoscale LCMS is done 75 micron ID columns, and what we're going to talk about is 1,000 micron ID column, and the relative sectional area is about almost 200 times bigger than that of a nano, and that comes with a couple of clear advantages. But just to illustrate why we even started this, you can see here two LCMS traces of human plasma digests, and the top one is the nanoscale and the bottom one is the microscale. And you can see very clearly that while we have a lot of problems here up here with the peak broadening, we can generate very, very sharp peaks using a microscale separation, and that gives peak capacities of around 700 within 45 minutes of LC time. So this simple characteristic led us to investigate this a little more, and essentially what we found for plasma is actually true for other body fluids as well. And what I should mention is, and maybe for the proteomics community that may come as a big surprise, we run the chromatography at 50 microliters a minute, which is a lot more than the typical 200, 300, 500 nanoliters a minute that you would do for a nano system. But you can see either for CSF or plasma or urine, basically all of these produce very sharp chromatographic peaks. And with CSF, we can get within half an hour gradient, 700 proteins, 250 for plasma, 1200 in urines, with loadings that are, for these body fluids really are not a lot. It's very hard to, even from a drop of blood, it's very easy to get a 20 microgram of plasma protein from a drop of blood. And what maybe is an even more important feature is the carryover from run to run almost doesn't exist anymore. So we have almost no carryover between runs. That is a great asset when doing proper chromatography. So, and here's basically a picture from the paper we initially published on this idea. When you look at the chromatograms down here, the one in red is a microscale LCMS, and in the blue it's the nanoscale LCMS. And you might appreciate that in a 15-minute gradient, the micro LCMS is already over before the very first peak on the nano system has come out. So we can easily double the throughput here with very little downside in terms of sample loading or also sensitivity. But this is certainly one thing to keep in mind, because we run at a much higher flow. The microsystem is about 3 to 10 times less sensitive than nano LCMS. So it will not be applicable to each and every application, but it will be applicable for a lot of applications. And this is what we try to show here in this table. Whenever we do full proteomes of cell lines or tissues, where you have 100 or 200 micrograms of total protein available, it should not be a problem at all to get to very competitive numbers of proteins and peptides compared to even often 2D LCMS approaches. So with that in mind, we also wanted to see if we can sort of stress test that system. And Yang came up with this rather elaborate way of putting together 1,500 consecutive injections that consisted of 10 cycles of 20 HeLa digests each, followed by some QC peptides, followed by 20 urine samples, 20 CSF samples, a couple of plasma samples, placenta samples, and then going around that circle 10 times. And essentially that was a total of 40 days of machine time. And we basically asked the question, how robust is this? And it turned out to be extremely robust. When you first look at the left-hand side of this plot, what you can see here is the elution times of synthetic peptide standards that we use as retention time calibrations. And when we run them alone, we get a certain, of course, retention time characteristic. And when we run them spiked into the body fluid, we, of course, hope that we will detect them at the same position. And you can see from this correlation plot where each of the dots here is a cloud of dots of many replicates of detecting these peptides, you can see that these two measurements correlate extremely well. So the slope of this is 1.0, so we're not getting any particular shift, and the intercept is 0.01 minutes, which is barely a second. So essentially that says the chromatography is so good that no matter whether we have a sample on the column or just a QC, it doesn't change the retention times of these peptides at all. And in the zoom in is shown here that even though these two QC peptides, they only differ in a few seconds of elution time, they are nicely separated by this system. And that chromatographic performance directly translates also into quantitative accuracy. So when you look at the coefficient of variation of the different types of samples we had in this stress test, you can actually see in this cumulative density plot that the vast majority of all these proteins is quantified with a CV of less than 10%. And that is, I think, quite interesting because it makes these measurements fit for many applications in the clinics, and something that is not really new to people who analyze plasma samples, but here are repeat plasma analysis of 5 individuals, and the t-SNE analysis very easily pulls those individuals apart, saying essentially that the plasma proteomes of individual people are different to each other. So we can now robustly measure this, and one of the so far unpublished stories is that, well, this is should also be high throughput, and actually it is. So we've gone through almost 400 cancer cell lines, and we're in the process of scaling that to more than 1,000 at the moment. And for each of those, we get in excess of 10,000 proteins for each cell line, and this is how they distribute. And for good measure, we've also looked at the phosphoproteomes, and you'd see that it is more variable here, but that is actually measuring biology. And here on the cluster heat map here, the only point I'm trying to make is that you can't really see anything, and you have to take my word for it that the 20 QC runs all cluster next to each other, essentially saying that we can now really cluster data according to what we measure rather than the device that is used to measure it. And that used to be a big problem. Okay, so we have good reason to believe that this is high throughput, but is it also robust? And that is something which we've just typed up again, and Yang will have that paper out in Analytical Chemistry in a matter of days now. So what we've basically, since we started doing this in 2018, we've kept doing it. And so what you see in this chart is here is the number of analyses that were run on this microflow LCMS system, either on an HFX or on a Lumos, over the course of about 2.5 years. And by now, I think we are standing at more than 38,000 runs and still counting. So that's good for robustness figures, and just here this pie chart gives you an idea of what types of samples these are. For the Lumos, we do a lot of high pH fractionation and read that out by LFQ quantification, but we also do a lot of TMT, and this is just for you to see how sort of these samples distribute. One really important factor in all of this, making it robust, is actually the lifetime of the columns. Even though some people may think for proteomics it's crazy to inject 5 or 10 or even 20 microgram of a digest, for these columns this is actually very little loading. And that translates into a huge column robustness as well. And this is shown in the pie charts here. So on our QHFX, one column has by now seen more than 14,000 injections on the same column, and on the Lumos it's 12,000, and both of these columns are still on the machine and they're still running samples. So that is obviously very good for any application where you want consistency in your analytical system. And if you have 5,000 samples to run in a project and you can do that on a single column, you're going to take out a lot of that variation that you usually have in the nano LC world, where a column can die any moment. And that is also illustrated here in this picture here. There's again these QC peptides that we ran sometime in 2019. You had nice separation of the two, and then something bad happened. There was a sample injected that partially blocked that column. So actually Yang turned it around, and we then started running it in the reverse order. And you can see that the retention time shifted a bit, but the separation was nice. And then more than a year later, almost a year later, he turned it around again to the forward direction, and the separation was still great. And now even the retention times returned to almost identical figures to what they were almost a year earlier. So injecting little into chromatographic columns is good for the lifetime of your column. It's good for separation. It's good for lifetime. It's good for data consistency. And this is just a different way of illustrating this here. Here is a couple of experiments using different gradient times from 10 minutes to 2 hours, run in 2018. And what is plotted on the y-axis is the full width at half maximum of the chromatographic peaks. And for 10-minute gradients, they are sort of the median peak width is about 3 seconds, and it goes up, of course, with longer gradients to about 10 seconds. And this is the same samples run 1.5 years later, and you can see that the distribution of chromatographic peak width is pretty much identical. And this is also the case when you look across single gradient, but many different, always the same sample, the QC samples that we run on HeLa Digest across a large number of runs across an extended period of time. So that really convinced us that this is also robust. And again, returning to the example of plasma, if you have a nano separation, it's very common to see these very sort of peak tailings on the late side of the chromatographic peaks. You don't have that on the micro columns, even though the loading here is 30 times different. And just to illustrate this again by zooming in, if you were, the blue line here is the micro separation, the red one is the nano separation, and it's not hard to appreciate that not only the blue peak is sharper, but also has less tailing. Okay, sorry for the bad quality of the graphics here. It's just a copy-paste problem, I guess, from a PDF. And Yang has also asked the question, well, can we actually use the system to go very deep into a body of fluid? And the answer is yes, you just need a little more time. So if you spend about 1 day of machine time, you can get to 2,500 plasma proteins or 5,000 proteins in urine. And he's just highlighted a couple of cytokines and chemokines that could be detected in this way, which are usually considered low abundance samples. So switching gears a bit, so we've convinced ourselves that that this system is both high throughput and also offers high robustness. And now the latest on this is still unpublished, but Yang also then asked the question, so what about doing proteomes at single shot? And a lot of people are trying to do this because, again, if you can simplify your workflow, including the sample preparation, the LCMS, the data analysis down the road, the hope is not only to be able to generate more throughput, but also better data consistency, because every step you take out from a workflow should lead to less variation. So what we've asked here is, well, how far can we push the idea of single shot proteome analysis? And the data that you see here in this slide is just counting the number of peptides and proteins as a function of injecting more and more and more digest into such a column, and measuring this either with a 28 or 41 Hertz mass spectrometer settings. And you may say this is a crazy amount of protein digest, but for those of you who are used to running Western blots, I mean, these are typical loadings here for a single well of a Western blot. And then you probe with one antibody or sometimes 2 or 3 to detect a particular protein. And if we do this with mass spectrometry, we can detect 30,000 peptides or close to 5,000 proteins that way. So that gave us some encouraging results that single shot proteome profiling might actually become possible. As a point of detail, you can see here that the slower mass spec method actually outperformed the faster, And the reason for that is simply that if you run the mass spectrometer too quickly, then you have too few ions that actually are being detected, and the quality of the tandem mass spectra goes down so that you can't ID them any longer. And we'll return to that in a moment. So what we then also looked at, obviously, again, is the chromatography. And what you can see on the left-hand side, as you load more sample digest onto such a column, what you observe is that the peaks get shifted to earlier retention times. This is not at all a surprise. This is what just happens if you have a lot of stuff loaded at the top of the column. So you basically get some displacement, but the peak shapes in chromatography are still very good. Another thing that you see is that at some point the whole system saturates. And this is not the chromatography, but it's actually the mass spectrometer. And that is apparent from plotting the actual ion current going into the instrument, and in red you can see the MS1 intensity really begins to saturate beyond loadings of 50 microgram. In MS you don't see that so much because obviously the ion current after fragmentation is always much lower than it is for the intact peptides. Okay, so we basically also what that says, loading more than 50 micrograms also really doesn't make much sense, and we're back to loading sort of Western blots with similar quantities. Now, where does that go when we now look at the gradient times? Obviously, one would expect to get more peptides and proteins the longer the gradients become. First of all, here on the left-hand side is a quick look at the chromatography. And these box plots basically, again, say how wide are the chromatographic peaks. Within 1 hour, we have about peaks that are 6 seconds sharp. And if we extend that to 6 hours, they become much wider. And in the inset is shown how the peak capacity develops as a function of gradient time. And we can see we can push it to nearly 1,000 peak capacity, but the increase from 1 hour to 6 hours in total is only about 50%. So one has to really think about how much time one wants to spend for a 50% increase in peak capacity. But what is interesting also is in the middle, when you again now look across the gradient times and ask the question, how many sort of precursors can we actually detect in MS1? We can see that is steadily going up. And how many of them can we try to fragment? And this is a relative scale always to 1 hour gradients. You can see the mass spectrometer is really trying hard to fragment many of those peaks. So it kind of triples the number of peaks that it selects for MS as the time gets longer. But the blue line here really is what is the result of this. There seems to be a sweet spot at around 3 hours where the largest number of peptides can actually be identified. So you might ask the question, so why does it go down again? And the obvious, the reason for that becomes relatively clear when you look at the right-hand side, where Yang plotted the precursor intensity of either those peptides that are identified in blue or in red, which they are not identified. And you can see that it somehow stabilizes here. You don't get any more identifications. And that is simply because the precursor intensities become too small for the peptides to fragment and produce enough ion current in MS to be able to lead to a identification. So that says going beyond 3 hours really also doesn't make much sense, because the mass spectrometer doesn't have the sensitivity in order to detect the fragments from those precursors that the mass spectrometer will still subject to fragmentation, but then the ion current in the tandem mass spectra is too small. Now, what does that mean in terms of proteins and peptides? Again, looking across the different times on this axis here, on the left-hand side for peptides and on the right-hand side for proteins. And now I'm bringing in also our artificial intelligence tool called Prosit that is rescoring essentially max quant results to tease out more peptides and proteins. And what you can see is as the gradients get longer, you also get more peptides, and you're losing a certain amount of peptides simply because max quant and Prosit don't agree that they are actually confidently identified. But when you look around on the protein side, it's actually quite possible from a HeLa digest, I believe this is within 3 hours, to get about nearly 7,000 proteins out in a single shot. So that's encouraging. And then we basically took this to some application where we looked at Arabidopsis tissues, in this case roots, the human breast cancer cell line, a mouse preparation of mouse small intestine or the thymus, and the story is kind of always the same. we actually get many thousand proteins, and the current record stands at nearly 9,000 for Arabidopsis roots. And that compares actually quite favorable to data from the Kuhn lab, where they coupled nano LC separations with FAMES gas phase separations, and in a single shot, also in 3 hours, managed to get nearly 7,000 proteins from whatever digest that they used here. So that is comparable to the figures that we're getting. So it seems that good chromatography can generate large numbers of quantifiable proteins. And again, I keep saying it, the micro-LC system is not as sensitive as the nano, but it's actually not that bad. Okay, last slide on this part of the story is just looking again at the quantitative accuracy, and the long and short of it is if we do replicate analysis of these different tissues, most of the CVs are below 20%, which again is good for quantitative precision. And then also if we ask the question, so what part of the proteome do we actually cover in a single shot? So what we've done is here just to plot out the abundance rank of these different samples, and then accumulate the amount of IBAC for getting an idea of how much of a particular protein contributes to the total. And then, of course, at the beginning, this sharply rises, and it's a typical observation that half of the total protein mass is contributed by only a handful of different proteins. But then you can see how this develops, and yes, there are some detailed differences in detail, but largely, essentially, this is the dynamic range we're getting, and you can plot this in a different way to get distributions of IBAC values, and you can see that the single shot method spans about 5 to 6 orders of magnitude dynamic range of detection. So that is again something that would also be very difficult to do in a single shot nano LCMS system, simply because you cannot load enough material. All right, so that was sort of the excitement around micro LC-MS, and my laboratory has converted at the moment 3 instruments in that mode of operation, and they are going through a very large number of samples, and we are not gonna, so we don't treat this as sort of a method development fun project, but we actually have made it an integral part of our analytical pipeline. So in the second pipeline we've just established, where we're following also the lead of other people, but with an interesting twist, is we have become more and more interested in analyzing clinical material that is formalin fixed, and that has traditionally been a bit hard to deal with, but a couple of improvements in the workflow that other people have made have really changed the situation. So for example, when you take macro dissected samples from a microscopic slide, of course, the first thing you have to do is decross-link this, and people have worked out protocols for doing that. Great contribution from Jeroen Kreiswerk lab was the introduction of this SP3 workflow, which allows us to work with things like SDS. And then for these clinical applications, we've basically settled on using this EvoZap LC system, simply because it has these disposable tips, so that we can deal with sample-to-sample carryover in a very pragmatic way. And what Yunjin and Stefan investigated, how this workflow could benefit from the addition of a FAMES gas phase separation in addition to the mass spectrometer. And so that's what they spent some time on optimizing, and on optimizing the sample preparation. It became quite clear that that having higher tris concentrations in the decross-linking actually helps, that keeping the pH under control helps in order to revert much of the cross-linking. And then maybe more interesting, because it's kind of newer, is then we looked at the distribution of compensation voltages and how many peptides we would be able to identify given either a gradient of about 3/4 of an hour or 1.5 hours. And you can see here the characteristics of those CV values, and you can use that information to in silico ask the question, which compensation values should you combine in order to maximize the number peptides you could detect, and here's just shown that the data matrix and the sort of the sweet spot in this particular case was 40 volts and 55 volts, which collectively should give about 30,000 peptides. But of course, this in silico analysis doesn't fully reflect the reality when you do the measurements. But nevertheless, when you analyze this systematically and ask the question, how many compensation voltages can we use within a single LCMS run, and how many peptides does that give us? This obviously is a question of how much time you give the system. Switching between compensation voltages takes some time. So with short gradients, it doesn't really make any sense to use more than one compensation voltage. And if you use two, it doesn't harm too much either, but you don't gain anything. If you use slightly longer gradients, you only really see a benefit the moment you put on two compensation voltage settings in the same run, but then also the benefit is over when you run 1.5 hours of gradients. Sort of you can afford to run 4 or 5 CV settings in the same run. And what I want to point out is that FAMES does cost you some sensitivity, because these data points over here are the runs without any FAMES, and you can see the moment you switch on one compensation voltage in FAMES, the numbers inevitably drop, but then they recover if you do gas phase fractionation. And what is true for peptides is also true for proteins. So we basically have settled on a system that uses, I think, 5 compensation voltage settings within a 1.5 hour run. And if we do this, and that's sort of the middle column here, we can cover about 40,000 peptides from this particular sample using a total of 2 injections of 1.5 hours each, and injecting 600 nanograms in each of those injections. And we've compared that to a classical basic pH reverse phase fractionation of such samples, which actually don't do so well, even if you use 10 times or 20 times more material. So it seems the losses here outweigh the gains. But then if you bring in FAMES to that one, you actually outcompete FAMES only. But again, you need quite a bit more material. So essentially for our purposes is we've settled on this middle ground here, where we get good coverage or reasonable coverage, but at very little total sample quantity requirements. And that helps us to deal with the clinical samples where sometimes there isn't a lot of it. And here's just to show on lymph nodes and prostates what the numbers are. And also there is some benefit from using the max quant match between run feature. One always has to be a bit careful how one uses this in order to not generate too many false positives, but that's been covered in the scientific literature elsewhere. So, and I think I only have 2 minutes left. If I look at my watch here, just two minutes on another. All right.
Go longer, yeah.
All right. So, but still, sort of third and last topic, which I'm only going to touch on briefly, is something we call the Proteome Tools project, where we have, together with the peptide company JPT and also with Thermo, analyzed more than 1 million synthetic peptides and generated hundreds of millions of MS spectra in order to generate essentially spectral libraries for many uses. And these peptides cover unmodified peptides, PTM peptides, endogenous immune peptides, and so on and so forth. And the idea really was to generate this collection of spectra one could use, we or others could use to learn something from. And it's very gratifying to see that somebody posted this a while ago, the list of 100 most downloaded projects from Pride, and the top #1 is Proteome Tools. So it seems people are downloading this, and that's of course great to see. And then we have a second tranche of data released, and in general, all of those arrows essentially represents work from my lab, and we do believe in data sharing for the benefit of others, and it's nice to see that people apparently use our data, so that's good. Sort of pointing you to the talk of Matthias Wilhelm later in the week is, what have we done with this? And one of the ideas was, can we learn from such data sets what the tandem mass spectra of peptides should actually look like? And here's an extreme case where this is this particular singly charged peptide that isn't tryptic, and it produced, from a synthetic peptide, it produced this spectrum. Sorry, that was the reference spectrum in the, yeah, so that's the standard peptide that we made, and in some experiment we initially found it, and the mass species among you might say this is a terrible looking spectrum, but actually when you make the synthetic peptides, that's what it looks like. And Matthias and people around him have built a deep neural network to be able to learn what those fragmentation spectra should look like, and we called this Prosit, and published this about a bit more than a year ago, and that turned out to be a really fun project because it looks like now we're looking at a triptych peptide, we can actually predict these spectra with a ridiculous amount of accuracy, and this is what is shown on the right hand side here. Is when you ask the question, how well do the spectra of the experiment and the predictions agree, then Prosit sort of gets it right with a median Pearson correlation of better than 0.99, and when we usually use the spectral angle as a measure because it's a little more sensitive, but essentially what it says, these triptych peptides can be predicted with an amazing level of accuracy. And the same is true actually also for retention times, and here's a well-known retention time model called SSR-Calc from Oleg Krokin's lab that really already a while ago showed that these retention times of peptides can be predicted pretty well, but Prosit does an even better job here. And so we've stopped putting more time into this because we think it's now kind of good enough. And that enables a couple of things. And maybe one of the most exciting things is that we can basically get a 10 to 100 times lower false discovery rate on actually identical data, simply because Prosit can rescore these tandem mass spectra with the question of, does the spectrum actually support the identification, and essentially using retention time where it should be, is it where it should be, and looking at the intensities of the fragment ions and asking the questions, is the distribution of intensities of these fragment ions as we would expect. And that is a very sharp knife together with a tool called Percolator that was generated by Lukas Schell's laboratory in Sweden to come up with these criteria that score identifications. And essentially the long story short is we can tighten up the false discovery rate requirements without losing much data at all. And that has advantages for a number of applications. For example, although I'm not showing it here, in the field of microbiome research, where you have thousands of different species together in one sample, so you have to consider a lot of potential genomes to search against, and this is where classical false discovery rate estimations really kill you, and tools such as Prosit can really help there. But more on that in Matthias's talk on Friday, but here ending just with another sort of gratifying observation is that when we put Prosit onto our public website, Proteomics DB, we actually saw that people began using it, and the fun fact is here, when you track that over time, the fun fact is here is somebody posted about Prosit on a blog spot post on Proteomics News, and look what happened. It seemed nobody had really seen this before, but when that posting happened, the number of predictions that were requested at our website began to rise sharply, and we also integrated Prosit within Skyline, and we can also see that the Skyline community is using this, and this I haven't updated in the last 2 months, but Prosit has made billions of spectrum predicted via Proteomics DB for the community, and Matthias has sort of converted this into, if you were to do this empirically, you would have to expend about 100,000 hours of LCMI's time to get those spectra. So that's nice to see that this tool is being used. And with that, sort of I'm coming to the end. I spent quite a lot of time on this micro LCMS system because I really would like you to consider doing this. It makes proteomics also more accessible to people. Nano has its problems and needs special equipment, whereas micro LCMS doesn't need so special equipment. I spent a little less time on the FFPE, but I think this is actually also a combination that works well, and others could of course too. And I didn't really spend much time on Prosit, but as I said now 3 or 4 times, you're going to hear much more of it later. And with this, I'm at the end, and I should extend special thanks to Yang Björn, who really made this micro LCMS possible, Stefan, Eckhard, and Nün for the famous work on FFPE, and Matthias and Tobias Schmidt and teams have made Prosit possible. And again, I have a wonderful team of people with whom I work every day, and they do all the work, and I get to talk about it. So with that, I'm closing, and I hope I had something interesting to contribute to your workshop, and I'm happy to take questions.
Mr. Kuster, thank you so much. It was really wonderful, and I'm sure very eye-opening numbers, and the kind of data which you showed from the micro LCs, I'm sure going to be game changer. A lot of people are going to adopt that and going to utilize the power of these chromatography to really build the deep proteome coverage. And I just want to tell all our workshop participants that many of them are independent investigators setting up their own labs for proteomics. I think there is considerable amount of time, effort, and patience goes into method development part, irrespective of whether you like it or not. Even if you are deep in biology, you have to spend still considerable amount of time in method development. What is best suited for your sample type and your projects, especially looking at the gradients, the run hours, and kind of the column usage. All of these things are unavoidable part of this game when you have to be doing this thing towards your project. And that's why I think a lot of time and intense effort goes into doing the proteomics. We are very also privileged to have on Friday Matthias Willem, who will talk about Prosit, and the workshop participants can definitely make use of the hands-on session which is going to conduct on this software. And you have seen the very beautiful numbers, how the community has adopted that and using that. With this, let me thank Professor Kuster for this wonderful lecture and open for the forum for the questions and discussions. Open to all of you now. All right, first hand comes from Dr. Sudarshan. Please go ahead.
Good afternoon, sir. It was really very interesting to hear you, and lot of data-based information. At many occasions, honestly, I was not able to understand also many of the things, but it was very good. I appreciate it. But 3-4 years ago also, we had heard some lectures from systems biology. They were also working on the importance of micro LC based in comparison to nano LC. So I wonder, and I would like to hear from you, that why the global scientific community now are more inclined to use nano LC based nano LC MSMs, in spite of all these good results from micro LC.
Yeah, so I think the breakthrough in proteomics actually came with the ability of dealing with what at the time was considered very small quantities of samples, and therefore you had to downscale everything in the sample preparation, but as well as the chromatography. And basically that breakthrough made a lot of people follow that lead, and then that stuck with people. But then there's, of course, and then it's hard to change. That is also true. When you've set up your lab to do nano LC, then and you've mastered to do this, then you stick with it, because going away from it at minimum needs another investment into equipment, and it needs an investment in time for people to try. But I think it's swinging back now, because particularly for applications where sample quantities are not a problem. And we are not the first to do this, really. We can't claim that we are the first, but I saw a paper in analytical chemistry, I think 3 years ago now, by Juraj Lenčko from Prague. And I was fascinated because he showed that these peaks are so ultra sharp, and you can actually do some meaningful experiments with it. So we can't claim that we've invented this at all. It's just we jumped on it, and it worked so well that we never will go away from it. But again, the DIA people have sort of walked away from nano a little earlier, because they also realized that chromatography is important. And some people I know who work with body fluids even use the classical 2.1 analytical columns, and of course they will need even more sample, but they're going to have even better chromatography. But for body fluids, it really doesn't matter. I mean, our protein plasma is at least 50 grams per liter, so that's 50 micrograms per microliter. So there is a lot of protein there. And also in urine, for example, 5 microgram of proteins from urine, I don't know, that's probably just 10 microliters of it also. So it's not a problem.
Yes, yes.
So although we, and last word maybe on sensitivity, in the Nature Communications paper, we did show that it is also fine for affinity purifications and also phosphorylation. But I will say there is this factor 3 to 10 in sensitivity that the nano has an advantage. And if people want to go to very rare PTMs or rare cell types, even single cell proteomes, I mean, there's no way that big scale chromatography is going to be the suitable method.
Yeah, thank you.
Great, wonderful. I think thanks for these comments. I'm sure participants are going to be very open who are all starting the labs that, what to go for, nano or micro LC. All right, Dr. Surekha Jingde, she is one of our proteomics scientist experts from India. She's next.
Professor Costa, I have two questions. How old blocks, FFP blocks, can one use for the study? And the second question is, how many sections do you really need to get reliable MS data?
Yeah. So the first question, I think, was about how old the blocks can be.
Yes.
We have not tested that, but I would imagine they could be ancient. The experience from a different area is from the gels, from protein gels. We analyzed proteins from gels even 10 years after the gels were run, and that was never a problem. But we have not tested this on the FFPE. But my gut feeling is it should actually be not a problem to run really old blocks as well. Then the second question.
Sorry, there have been concerns about how the material was fixed within the paraffin and what paraffin was used and so forth. So these are the concerns that come up.
Yeah.
And therefore, the aging also would matter.
It could. I haven't got any data on this. As I said, it's just a gut feeling. The second is how much does one need? The data that I showed, I think, were performed on 4 slices of 10 micrometer each.
Okay.
But then it usually generates 10 or 20 microgram of protein, and then we only use one of them. So I think it's entirely possible for macro dissected samples to use a single slice. Let's say 5 by 5 millimeter is, I think, our standard. We haven't tried tissue microarray type of samples. That's on the list of things to try. That could become more challenging because they are much smaller. But yeah, just for reference,
it depends on the size of the tissue in the block.
Also, of course, yes, and the heterogeneity. What we are also only beginning to learn, of course, is the tissue heterogeneity within a block will, of course, have an influence on what you get out from the proteomic experiment.
Correct. So everybody starts looking for single cells or different types of cells within that tissue.
Yes, but that is something that is going to be still hard for some time.
Yes. Yeah. Thank you so much. Yeah.
Great, thank you. So now next question is from Argyo Banerjee. Argyo, go ahead.
Yes, so it was a nice lecture. I will come to the point. According to the slide, what you showed in Baker Jensen et al. Okay, you have taken the Prosit scores of 1% FTR for identification of the highly confident peptide. Now, each of these identification of the confident peptide depends upon like what factors, what variables you use. If I am going with the Orbitrap system, like Orbitrap kind of system, people say that you have to match it with the NIST library, like the MS Pep search and all that. You have to match with the available synthetic library for the collection of the Orbitrap, and if it matches, and if it has a score of less than 500, means more than 500, or an correlation score of more than 1, then you can tell that it is like good, like your peptides are highly identified, your peptide is. I have seen a score of here also, like the Andromeda score and the Prosit score. So like as you have compared with the MaxQuant, like how your software is robust with the MaxQuant, have you tried with the other commonly available search tools like means Proteome Discoverer, Spectranaut and all that? And how is the sensitivity of your software as compared to all of those? Means then it will be a great revolution of proteomics, because everybody is able to defend their data by their own side. But when we are running the raw list parallelly, we see that some peptides come in sequestest, some peptides come in Amanda, MS Amanda, some peptides come in, means they are not, like, it is not occurring throughout. This is the ambiguity.
Yeah. So clearly there is ambiguity, even if you filter by whatever criteria, 1% is still 1%, and and 1% of 100,000 peptides will mean that 1,000 peptides of them could be just there for random chance alone. And all of the search engines work using kind of different and similar ideas. So I don't know exactly what's under the hood of MS Amanda, and I don't know exactly what's under the hood of MaxQuant. But essentially what we do with Prosit, actually, it's not a search engine. We are taking the results of a search engine and ask the question, does that identification that the search engine already made, is that plausible or not? And for that, we use the predicted and the experimental tandem mass spectrum and essentially compare them together along with all these scores provided by Percolator to come up with essentially a yes or no, whether or not they are within our 1% criterion. So to answer your question more directly, what frequently happens is that the rank 1 peptide of MaxQuant will not be the right one. We promote often the rank 2 because the spectral actually are a better match, and MaxQuant gives more weight to mass accuracy, but it doesn't use the intensity of the fragment ions. We have, together with Thermo, integrated Prosit into Proteome Discoverer. So that's something that all the Proteome Discoverer people can now use. And the results of Proteome Discoverer searches, or what under the hood of that is Sequest, is not identical to MaxQuant. And therefore also the rescoring results will not be identical. In that sense, the idea of uncertainty will always be there, but what I try to say is by extending the capabilities of search engines to the question of, or to the using the information of retention time and relative fragment ion intensities, this adds discrimination power. And it adds actually a lot of discrimination power, so that even if we didn't want any more peptide, we could tighten up the stringency of the FDR by a factor 10. Or conversely, if we still think that 1% of error is okay, then we're going to increase the number of things we have in our box.
All right, great. So these are really, very insightful comments. And just to convey to all the participants who are, some of them are new to proteomics, there are different type of softwares available. MaxQuant and Proteome Discoverer, some of these are very popular software. MaxQuant being open access and Proteome Discoverer is a commercial one. But fortunately, in this course, we are going to show you both of these software and how to analyze the data. In fact, it will be in your favor to try to take the same data file and analyze using both the software and do compare what we just discussed, that you will see some differences in the output, and then you can appreciate the kind of differences these algorithms have. Any other question any participant has? Any last question?
Just a last question, like this Prosit can be accompanied, means it can be installed with the Proteome Discoverer after Percolator, like after the validation, correct? So we have this third-party installer.
Come again?
So like if I want to incorporate this Prosit in Proteome Discoverer, so we have that options available.
So I believe version 2.5 has it on board.
Okay.
Great. All right. So with this, let me thank Professor Kuster once again for giving this wonderful lecture and showing you how much hard work lab goes into optimizing the method before you see those beautiful papers out, revealing the biology, and these valuable comments from his experience is going to be quite helpful for all the participants who are gearing up for doing proteomics in their laboratories. So Professor Kuster, thank you so much from the organizing committee of IIT Bombay and all of us in this workshop for delivering this wonderful plenary talk. We are very grateful for you, and we look forward for further session from Matthias Willem from your group on the Prosit. Thank you once again from all of us.
My pleasure, and have a nice course.
Thank you. All right, bye-bye. Great, all the participants. So now we will be doing a quiz shortly with all of you. So I hope you're all ready. we have given you some data file from analyzed